Please join me in welcoming Melanie to the stage. So, what lurks below? Let's have a look. Tonight's Antarctic adventure is going to be about my Larsen C. Benthos expedition, which was in February and March this year, for about a month. I'll be talking about where we went, why we went there, who was involved, how we got there, life on a ship, science at sea, and then of course some ice and animals. So I hope you enjoy it. But first, a little bit of background. So this is going to be about my day job. So for those of you who don't actually work at Museums Victoria, I work behind the scenes in the collections here and help to manage a beautiful collection of marine invertebrates. We have over 1 million specimen lots, so over 5 million specimens, and these are accessed quite often, every day actually, by everyone from students to international scientists. So they're a really important life library of animals. But my special interest is in holothuriids. So holothuriids are sea cucumbers that come in all shapes and sizes, from these shell-like solids to the squishy sea pigs and everything in between. And they're actually related to echinoderms that you might know more, things like sea stars and sea urchins. Now, sea cucumbers have not always had the respect they deserve. Even in 1908, they were being called plug ugly and disgusting and repulsive. And to be perfectly honest, every now and then, when you see pictures like this of preserved specimens, they don't always look that pretty. But they are amazing animals, I promise. So lots of different things about sea cucumbers. They're ancient, so over 400 million years they've been around for. They're the earthworms of the ocean, so they do help churn that seafloor for everything else. They've been used as a medicine and a food source in many different cultures. They're also Australia's first export, and I'm talking about at least 300 years before Cook even saw Australia. They use hydraulic pressure for locomotion, so actually to get their tube feet working and also to get their tentacles working. They can breathe through their butts. They can vomit in self-defense, shoot predators with sticky toxic tubules, and they have some amazing ways of reproducing, everything from spawning to splitting in half and just regenerating. They're also great habitats for other animals. Some of them have worms living in association with them, or gastropod snails living inside them, and fish living in their butts again. And there can also be taxis or Ubers of the ocean too. I've seen some fantastic footage of crabs hitching a ride on the back of sea cucumbers. And some even carry the young in a pouch on their back, like this beautiful picture from the Falkland Islands. And they're also gorgeous on the inside. These are ossicles, so these are calcium carbonate, and these are in the tissue of sea cucumbers, and we use them basically like a thumbprint to help describe a species of sea cucumber. And sometimes they're even cute on the outside. So this one has inspired that lovely little plush toy down there, the sea pig. And a tasty treat for other animals. So you can see here that it's sometimes they're relatives that are the nasty ones. And you guessed it, they've even been found in Antarctica. But many of those Antarctic sea cucumbers have actually been described here at Museums Victoria, and that's where the link comes in. I've been lucky enough for many years now to work with Honorary Associate Mark O'Loughlin, who is in the audience tonight, with quite a few different people on Antarctic and other sea cucumbers. And we've worked on a lot of the historic expeditions, things like the Discovery Expedition, the Terra Nova Expedition to Antarctica, as well as the more recent ones from the British Antarctic Survey, New Zealand groups, American groups, and things like this. So with all of that knowledge, it was what got me on another trip south. So this time, my recent field work to the Larsen C in Antarctica. So as a bit of an overview for those of you who don't know much about Antarctica, it is the coldest, driest, windiest continent on Earth. So I'm talking about that it can get down to -89, which is absolutely freezing. Luckily, we didn't get that. There are katabatic winds that roll off the mountains and come to hurricane speeds. And one of the really important things for tonight is that the snow rarely melts there, so instead it sort of just piles up and starts to compress and form these glacial ice sheets. And they can be nearly 2 kilometres thick and have about 70% of the world's fresh water, so that's an amazing amount of water that's held in those ice sheets. So back to the where, this area of Antarctica. I like to think of it as the Rhino Horn, also known as the Antarctic Peninsula. And for some historic context, this is actually the area where Shackleton's endurance ship got caught in the ice, started to drift and eventually got crushed and sank. Why did we go? A not so little iceberg called A68, unfortunately not called Bob. So for the background, in July of 2017, so just last year, a huge iceberg about 4 times the size of London broke off from the Larsen Sea ice shelf. Now, this whole area had been covered since the last interglacial period, so there was a call for a rapid response survey to see what was under there. If you have a look here, you can see the aerial view of that iceberg splitting away from the Larsen Sea shelf. And this is one of my favourite explanations of size. Apparently that's about 85 elephants high. So, and first exposure for hundreds to thousands of years, if you can imagine that it's like animals that live in a cave. So they're in this dark, cold environment. Maybe they don't need eyes, who knows, they don't get a lot of food. And then all of a sudden, the top is lifted off and they're getting all this new food and light source coming in. So how rapidly is that going to change? And what we really need is a baseline to figure out what will change and how fast. So our concern, of course, is that this kind of thing is going to happen a lot more with climate change. And so our aim here was to use this as a test case to establish that early biological baseline. What's living there now? Is it a unique ecosystem? And what changes over time and how fast? Now to the who. So I was very lucky to be part of an international group of scientists on board here. There were 29 officers and crew and 25 in the scientific party, including marine engineers and a couple of media people too. Scientists on board from many different institutions. It was actually led by Dr. Katrin Lindsay from BAS and funded by a NERC urgency grant. And of course, I'm very grateful to the MV team for letting me go and for BAS for supporting me as well. So how did I get there? You'd think you'd just go straight down, wouldn't you? No. I went south via north. So it was an urgent rapid response survey, so I found out about the opportunity quite late in the day. And by the time I went to buy tickets to Chile, it was too late, they were already all gone. There's only 1 flight a week down to the Falklands, so the only other way I could get there was via the UK. Now, I also needed my gear anyway, so it actually turned out to be quite convenient. So I flew into London and then went to Cambridge, where BAS is located, the British Antarctic Survey. I had a kit fitting so that I could get those layers and layers of clothes and hard hats and things like that I would need. And then I did my sea survival training in Norwich, so again, up there. And then back to Brize Norton to catch the Royal Air Force plane all the way down off the coast of Africa and down to the Falkland Islands. Another adventure which I won't talk about here. And the Falkland Islands themselves are known to many people as a gateway to Antarctica, and they really are quite beautiful. They've got their own stunning scenery and some really amazing wildlife, but that's a story for another day. What I was there for was to join this ship, the James Clark Ross, gorgeous red icebreaker. So for a little bit of a tour of the ship, I'm going to show you around these different areas. The scientific hold is where we stored things like jars for pickling specimens, any extra equipment and things like that. It also had access, the roof could be taken off so that cranes could be lowered in there, so anything that was really heavy could be stored down here in the scientific hold. This is our main workplace, the aft deck. So we spent quite a bit of time out here and then also in this lab, which was just directly off that deck. There are also several other labs, and for each of these labs we had to climb through watertight doors to get to them. So you can imagine it can be quite difficult at sea when you're carrying lots of specimens to climb in and out of doors. Monkey Island, so you'll see from the picture at the top, this is about as high up in the ship as you can go. It's also a fantastic place for whale spotting and things like that. And you'll see there that people are smart enough to stand behind that so they don't get too cold. So there's a lot of wind up there as well. The bridge, of course, where the captain is, and he's probably pointing at a giant iceberg coming our way. And one of the best views is actually just from the front of the ship there. If you're wanting to crunch through ice, it's a great place to watch it. The UIC is actually where a lot of things happen. So whenever you're not sure what's happening in the day, you go straight to the UIC. And you'll see over here we've got the winch operating here. We've got all the information about temperature and where we are. We've got some underwater footage. We've got maps. So a lot going on in the UIC. And of course the officer mess. Now this is what sort of makes it a little bit different from an Australian ship, I guess. Mostly the picture of the Queen and Prince Philip over there. They've even got lovely monogrammed carpet on the floor. And we actually even got menus, service, white tablecloths, everything, unless there were sticky mats required. So sometimes it actually is a little bit too rough to use a white tablecloth. And some very interesting adaptation of sausages. There's also a lovely lounge, which is a great place to relax at the end of your working day. And we had a knitting club going on in this one, but as you can see, people have even made up their own Monopoly boards. There's a dartboard, which you can imagine that's not always easy at sea to use a dartboard. Never stand behind that one or to the side. And one of the most important things, the fruit bowl. So for those of you who haven't been to sea before, if you're on a longish voyage, then the salad, sometimes the eggs and the fruit are the first things to go. So it's not the sweets, go straight for the fruit. You really need it as much as you can at the start. There is a hospital on board with Dr. Pips there. So she does even have an operating table. So, if anything was to go really wrong, she could help out. And down in the forward hold, there was a bit of space there. So it's really important at sea also to have that work-life balance. You're in a very enclosed space with a lot of people. So it's very important to try and exercise whenever you can. And luckily we had a purser, Richard, there, who was very happy to train us, get us going on some circuits, and also the doctor who was taking some yoga classes too. So whenever we had spare time and the energy, you try and fit in a little bit of that as well. And they did have a gym and a sauna too. Tiny, but good. The cabins were actually good. They're small, but comfortable. And then we started heading south. So just to give you an idea, from the Falklands there, I don't know if you can see that red line, all the way down to the Antarctic Peninsula. So down to that rhino horn. So to do that, you have to go through the Drake Passage. The Drake can get quite rough. Yes, we were lucky. It wasn't too bad this time, but every now and then you'll get a big wave or a big roll. And then you get to the calm crunch of the ice fields, which is lovely. So, of course, negotiating life at sea. A lot of people do get seasick, so particularly for the first few days, it's always important to either have seasickness tablets with you, or for me, I tend to stick a little bit more to ginger nowadays. And you do have to really be prepared. There is that old saying, one hand for the ship, and you really have to always be holding on. You don't know when that roll is going to come. And even in a cabin that's supposed to be sort of sea ready, if you have a big roll, everything can fall over like it did here. And you'll notice just here, an immersion suit shoved under a mattress is a great way to prop the mattress up against the wall too, as is this anti-roll device, so that you don't fall out at night time. And you can see in the shower here, things like handles and those slip mats on the floors and on every surface are really important on a ship. Blackout blinds. Now for this trip, we didn't get far enough to have 24 hours of light, But blackout blinds are quite important even without that, because at night time a lot of seabirds are actually attracted to the ship lights too, so it's good to have all of that closed off. You'll see here, so this is a cabin that had just 2 beds in it here, and you'll see we've got humidifiers, makeshift humidifiers up there, so that was soaked in a little bit of eucalyptus oil. The air gets really dry in Antarctica as well, so you've just got to think about things like even just being able to breathe normally, and how dry your skin gets as well. And of course, it's very hard to get your own privacy on the ship, but luckily there's these gorgeous curtains here if you needed some alone time. And of course, coping with the cold itself. So I said before that when you get kitted out, they give you layers and layers of clothing, and sometimes it's sort of up to 7 layers that you might be wearing in a day when you think about all the thermals and that you've got going. For this trip, even with gloves, I was really layering up. I started off with just a thin layer of gloves with the surgical gloves over the top, and then another layer over that, and it was still too cold, so I ended up having to use antifreeze cream under all of that, just so that my hands could cope with even 10 minutes out on the deck. So where we were headed. This is a satellite image. You can see there where the iceberg's broken off. And our plan was to try and get down and follow all these leads here, get all the way around to this area. Now it seems like it would have been easy to go up the top, but as you can see, the iceberg wasn't really doing what we wanted to do and kept bumping back into the sheet. And it was also starting to crack off, and it really just wasn't worth coming through this way because we could have got hit by huge bergs if they came through. So it was easy going at first, and I'm just going to give you a bit of a sense of being there by getting a video of moving through those leads. So that's what it's like when it's easy. Still very cold, you can sort of get a sense of it from that wind and everything there. And then it started to get a little bit tougher. We hit a lot of pack ice, and this was constantly moving and refreezing right into our planned track. So the pack ice is formed when sea ice just joins to other sea ice, and then it just gets bigger and bigger and bigger, and this can be multi-year, multi-layer. And it does move a lot with the currents and the wind, but it's really hard to get through. And you can see there how thick it can really get. And sometimes, even looking out the portal, you could see nothing but ice. So it was really starting to close in around us. Yeah, you can really see it there. So that's just the view from the deck. So as it was getting tougher, these are the kinds of sounds that we started to hear. It actually was much, much louder if you were down in the hold as well. So you can just imagine hearing that scraping ice against the hull and just thinking, is this okay? And we started to require a bit of a run-up. So the thing with an icebreaker is it can use power as much as possible, but at some points it's just still not going to be able to get through that ice. So what they do is they run up, try and slide onto the ice and use the weight of the ship to crack through. So this is what the crack looks like when you jump on there. And you can see there, creating our own leads with that. So it's a great idea as long as you don't get stuck on top. It's all right, we didn't. But I have heard that they've had to use cranes and that in the past and move huge containers around on the ship just to get off an ice floe if you get stuck on one. And then the wind and the weddell jar just got in the way. So we did this for days trying to get through this ice down here. And you'll see here this giant weddell gyre. Now, the thing about summer in Antarctica this year was that it was actually, a lot of the ice was melting in different places and a lot of it was cracking off and moving around. And a lot of it from over this side, where the Filchner Trough and everything is of the Weddell Sea, that area has been ice covered for a long time, and all of that ice started to break up and, you guessed it, moved right into our path over here. So even though it's one of the most ice-free summers ever in Antarctica, we got all of it right in our path. So the sea ice itself was 4 to 5 metres thick, and we had been going probably sort of about 8 kilometres in an hour, and instead we were reduced to about 8 kilometres in 24 hours. So we still had about 400 kilometres to go, and at that point we realised we just weren't going to get there in time, and even if we did get there, it was likely that we were going to get stuck and have to overwinter in the ice. And we didn't even have a helicopter, so that was not going to happen. So it was get out or get stuck, as you can tell from this night shot here, completely surrounded by ice. Very frustrating, but also a very good decision. And as the great man himself once said, difficulties are just things to overcome after all. So where we went, take 2. Okay, so we didn't get down quite as far as we wanted to, but we still wanted to get a lot of science done. So we looked back at this area called Prince Gustav Channel, which was up near the tip of the Antarctic Peninsula. So where we actually got to was about down to here, and we just couldn't get any further. So we went back up and around to look at this area. Now, this area hadn't been sampled for science, for biological science before. It had been sampled a lot for other things. It's a stunning area, and it was also covered by an ice sheet up until the 90s, as was there were 2 different ice shelves covering the area. There were some areas there too that were potentially ice-free for much longer. So it still gave us an opportunity to add to that story about what happens after an ice sheet is reduced or after something breaks off from an ice sheet. And it was just beautiful. And just to give you an idea of the depth, it was from about 200 metres up around here down to about 1,000 metres here. And that's James Ross Island. And we're, of course, on the James Clark Ross. So again, we were looking at setting a benchmark of seafloor biodiversity, so it hadn't been sampled for this before, and also looking at a more accurate time frame for those retreat of ice sheets in that area. Science at sea. So some people do dive in Antarctica. I've never tried it myself. I'd love to, but it looks very cold. But what we wanted to find was at the bottom of the ocean, so that's way too far to dive and of course too cold to get there. So how do we collect animals? We use different collecting equipment. I like to think of it as basically mini fishing, scientific fishing. We use the AGT, the Agassiz Trawl. This is used for bigger animals. You can see it's very sturdy. It's about 2 metres across and it has very coarse fishing nets. And we tie knots in the end of those nets, drag it along the seafloor and bring that up to the top. You'll see here, I don't know if that works. No, not working. There's a bit of a dent at the front there and you can see where it's actually hit a rock in the past. The EBS is used to collect smaller animals, and they get captured in these finer nets and taken all the way into those cod ends, which, yes, there you go. You can see the cod ends there, which are not so big, so it's only about this much. And that's what they look like when they're in action. So they're both in retrieval mode there, coming back up from the seafloor. You can see those laser lights at the front with the camera. And this is what happens as it gets dragged along the seafloor. You can see all those little dents and holes in there from all the rocks that it's hit. And that one from a giant rock that it's hit. There was also a CTD. So this is for water sampling, and people were using that to test things like temperature, density, things like that, but also to collect water that could be used in experiments on board. And the multicorer. So this one gets dropped down and basically takes a core, like an apple core, of the mud and brings up, hopefully, animals and sediment samples, and an unfortunate brittle star there that got stuck. There are also these bongo nets. So this was collecting at the surface and looking for plankton. And we use the SUCS, the Shallow Underwater Camera System. It looks like a little tripod here, and that's basically all it is. It's a little tripod that we pick up and move along the seafloor to see what's there. And we actually direct that from up top. The poor winchman actually has to sit down on deck to lower and move it with a joystick, but at the top we can sit in the UIC, take photos and talk to the bridge and talk to the winchman to tell them when to move along. And finally, the last piece of equipment is a good set of binoculars. We did have people observing mammals and seabirds. So a typical day of sampling. First thing when you get up, if you've had a bit of food, is to, of course, go to the UIC and check the whiteboard to find out what the plan for the day is. Now, I should just say the first plan for the day. It often changes. There are also people swathing the seafloor here, so that's when they ping sonar off the bottom of the seafloor to map it, to find out what trenches are there, what mountains are there. Basically, a good place for us to put down the equipment and to see how deep it is. And then we deploy and retrieve the equipment. You see there, every day is leg day. It's actually, quite difficult and very, very heavy to retrieve this kind of equipment. And if things like the cores come up, if they're successful, then of course they got sliced off and sampled. And that's those cod ends from before, from those finer nets in that sled. And you can see that just pretty much got pickled immediately because there are just so many, probably hundreds to thousands of animals in there, tiny little animals, that we needed to get them into ethanol straight away. Larger animals we started to pick off the net. So you untie the net at the base and put that into a bucket or a container, whatever you can, and take that into the lab. But a lot of the animals get tangled up in the net itself, so it's important to get off everything you basically can. And sometimes you hit some mud as well, as poor Thomas did there. So if you do hit mud, we need to sieve it on the deck using seawater there, and then ferry those animals back into the lab, where of course they're sorted. Now when I say that they're sorted, you're basically just starting with like. So if something looks like a sea star, you're putting it with everything else that looks like a sea star. If it looks like a worm, put it with the other things that look like a worm. And then you go further from there. Everything was photographed, counted and preserved. When we preserve things, we put it into ethanol. Often down here we're using 95% ethanol because the seawater itself in the animals was diluting it. And it's good to keep the ethanol at about 70% or more. And also 95% is great for DNA, so people were sampling for that as well. And then, of course, we needed to label and database and transfer the specimens to the freezer, so they stayed as well preserved as possible. If time, we identified them further, but there wasn't a lot of time because we'd spent so much time at the start trying to get through the ice. So we were really looking at a rough sort here, and the specimens themselves are actually currently, I think they've just arrived back in the UK. They stayed on the ship, and now they'll be sent back out to all the different museums for people to keep identifying those animals, perhaps for years to come. There's some databasing. And something that I kept forgetting to do is to look up. So when you're working hard in the labs and everything, sometimes you forget where you are. And when you're in Antarctica, it's really worth looking up now and then because you get this kind of thing going past. Now, as I mentioned earlier, things don't always go to plan. So this was about contingency plan 3 or 4, I think, by the time we got up to Prince Gustav Channel. But even there, still a deployment can fail. So the wind can be too strong. The bongo nets actually needed to be with light wind to go out. The winch broke a few times. If you hit rocky substrate, you can get all tangled up like this. I mean, there's so many things that can go wrong at sea, and I think anyone who's ever been to sea can tell you that's just a normal day. There can be too much ice and too many rocks. So if there's too much ice, the problem is that there's not enough space to actually deploy the equipment, or the ice has moved into the path you've just gone down. So what you really want to do is maybe have a look with a camera first, see what it looks like, then go back to the start of that transect and do it again with a sled, go back to the start again and do it with an AGT. So you're kind of wanting to go over similar or right next to an area so that you're getting a proper sample of that space. But if the ice moves in front of it, what are you going to do? Now this here, you saw that big hole in the, big dent in the trawl before. These are glacial rocks. So these ones are from the Falklands, and you can imagine these get pushed out with the glaciers. So it's basically like an extremely slow-moving river of rock and ice that gets pushed down towards the ocean. And a lot of those were in our path, but also they can get picked up by icebergs and taken out onto the water and dropped as drop stones too. So we had to always be very careful to avoid these so that we wouldn't lose our equipment altogether. And sometimes everything just freezes. So we had animals freezing to the nets as soon as the nets landed on deck, which I've actually never seen before, and this is my 3rd time in Antarctica, so it was really cold. The seawater hoses froze. Great. So you need seawater when you're sorting these animals. You might be sorting them for hours, and you actually need to keep them as cold and fresh as possible. So when they freeze, then you have to fill buckets from the fire hose and cool those down in snow. The CTD water froze, so again, that water that was coming up in those bottles and used for many different experiments was freezing, and people had to wait for it to defrost before they could do their experiments. And of course our hands. But sampling was achieved, science was done. We actually were very lucky. We got through quite a few deployments of all the different equipment, from 200 metres all the way down to 1,000 metres and more, so really quite deep. And this is the cruise track. So you can see here, this is the Falkland Islands. And we wanted to get down here. This is as far as we got. So you can see, yeah, that's like, it's 400 kilometres. There was just no way we were going to get through, unfortunately. And when it all does go to plan, what do we find down there? Lots of different amazing animals. Now, I've just brought a couple of them along today, which I'll just say, if you haven't been to the museum behind the scenes before and seen any of these animals, feel free to come up afterwards and ask me about them, and I'll show you some of the critters that I've got down there on the table. But we got so many different kinds. You see here this beautiful pycnogonid sea spider. So sea spiders are not related to land spiders, and this one here is probably about that size. But the sea spiders in Antarctica are all different shapes and sizes. You can get really small ones as well. And you can see there that it's got a proboscis, so a straw that it uses to feed, so it can't bite you, so it's okay. Some Comasians there. This is not to scale, by the way. These Comasians are actually only about this size. They're tiny. A great food source for whales and things. We've got, of course, a few sea cucumbers in here. Things like snake stars, these guys are gorgeous, related to brittle stars, and they like to curl themselves around things like coral. And you'll see here an urchin, a pencil urchin, and on the spines it has a whole lot of different bivalves, so little clams, and it also has sea cucumbers too. So there's not a lot of food down here in this deep, dark, cold area. So any chance you've got to get further up into the water column to get more chance to eat, you'll take it. So you can see these guys are competing for space at the tip of the spines so that they can get more food. And I do have one of these down here, so you can have a look later if you'd like. So different kinds of seafloor meant different kinds of animals. We saw Ophiuroids, brittle stars, everywhere, these guys here. So they were in a lot of different places, but particularly you saw them in the muddy areas. And you can see sort of holes and things everywhere. So you've got a lot of worms and that living in here too. A giant sea cucumber there, that one is about that size. And anywhere that there's, see this is a stone here. You'll see anywhere that there's a drop stone, there's often a lot more animals because they can attach to something. So there's some feather stars stuck on there. So here's the rocky substrate, and you can see there's a lot more critters down there. You'll have corals and sponges and things growing off them, and there's another sea cucumber propped up there. Really pushing the sea cucumbers, aren't I? Lots of other ones there, and lots of worms and things like that too. And the drop stones that I mentioned earlier, so bad for equipment, fantastic for animals. So you can see on here, these are not plants, these are all different animals. We've got corals, we've got hydroids, we've got bryozoans, and those then themselves become habitats for lots of other animals. Sponges, also a great habitat for other animals. And alien-like amphipods, I love this guy, I look like a little punk. Might even be a new species. We had an expert on board and she was very excited when she saw this one. And these great isopods too. These ones, probably about this size, which is quite big for an isopod. Isopods in your backyard are things like pill bugs and slaters. So they look quite different. Big worms, that's the technical term. Little worms, also the technical term. Lots of different worms. We had 2 worm experts on board and they were pretty excited and pretty busy the whole time. And sea spiders, like I mentioned before, and you can see here the different kinds. So you've got one of those big ones there, but then all these little ones with lots more hair on their legs. And there's actually probably at least 5 different species just in that tray. Beautiful amphipods, so many different kinds. And cerolids, I like these guys. They look a bit like trilobites, but they're not. I've got one of these down here if you'd like to have a look later as well. And of course, echinoderms. Gotta love echinoderms. So again, related to sea stars, there were sea stars down there, but also a lot of these brittle, basket and snake stars, which are really just beautiful. And sea cucumbers. More sea cucumbers. So this is not to scale again. You can see these guys are quite gelatinous and they've got this little vellum here that they use to swim and they just sort of move like this. Gorgeous sea pig right here. This guy's actually really quite small. You can see there it's using that water to push out its tentacles, its feeding tentacles here. And see these little white spots? They're actually clusters of those ossicles that I mentioned at the start, those little like wheels and hooks and things like that. Now, fortunately, we saw mostly animals, but I do have a video message here from Susie to tell you about what else we saw.
We're here on the James Clark Ross and we're sampling animals on the seafloor in this amazing Antarctic icy environment. We've found some incredible life, lots of diversity down there, but sadly we've also found so far only one piece of small plastic that we pulled up in one of our samples. This looks like a piece of plastic cable. It's the only thing we found so far, but we are also taking sediment samples of the mud down on the seafloor, and when we bring these home, these will be analysed to see if we find any other traces of microplastics or plastic fibres in these samples. Hopefully we won't find anything else and that the rest of this environment will be as beautiful and pristine as it looks from here.
So, so quite different from the Abyss trip. We were finding a lot of rubbish on the seafloor because it was in the shipping channels around Australia. Luckily, so far, Antarctica is looking much better, but we know from people who have been doing a lot of research on microplastics that it has been found down there, unfortunately. So we were taking samples to feed into those projects. So now just a few happy snaps for you of some of the ice and the animals that we saw from above the ship as well. So one of my favourite things about Antarctica is just how beautiful and how different the ice looks wherever you go. So you can see here it's just starting to form, and you can get these beautiful lily pads. Just really gorgeous, this pancake ice as it bumps against other pieces. And just there you can see it's just starting to form sheets on the surface. And then they're starting to layer up on each other, really quite stunning. And on a nice sunny day, beautiful blues and whites. So you think of Antarctica as being white, but it really is blue and white if you're in the watery areas. Gorgeous ice caves and things. And of course, huge icebergs. So you might only see 40 metres, but you know that there's a lot more below. As I said before, 85 elephants high. And you can see here, this is just before the water started to freeze, and it creates that almost mirror. Absolutely stunning. On an earlier trip, I'd seen some whales come up through this water, and just the way they started to edge through, it would hold for a good few seconds before it broke. So you can really see that ice starting to form. And the icebergs themselves have their own, almost their own climate on top. And just lovely seascapes there, particularly in Prince Gustav, but also all the way down the peninsula. And we were really lucky in that channel area that we were surrounded by glaciers and cliffs, and when the cloud cleared, it was just breathtaking. This is one morning we got up and went to Monkey Island and saw these little footprints belonging to a stranded snow petrel. But luckily, we had someone on board who could handle it and get him off again, and he did actually fly away, which is, they're not always that lucky, the birds in Antarctica, but this one got away. There was some sneaky skuas sort of circling around trying to keep an eye out for him, but I think we didn't see him get caught, so we're hopeful. Lots of different birds. I don't know all the species names myself, but we did see shags and terns and gulls and petrels and shearwaters and skuas and fulmars and albatross, and lots of different species of each of those as well. This one here is the nice goth one, black-browed albatross, one of the ones you can tell. And whales. Didn't see as many as I was expecting, but I think that's just because there was just so much ice that they couldn't get up to breathe where we were. This is a humpback. Lots of Adélie penguins. They were the main penguins we saw. We did also see chinstraps and emperors and Magellanics. The chinstraps are quite cheeky. They like to come up and have a bit of a chat. And the seals, we saw weddels and crab eaters and leopards and Antarctic fur seals. So there's that black-browed albatross. And this one's a wandering albatross, and you can't really tell from the picture, but sometimes the wingspan can be up to 4 metres across, so really huge. You're looking at them as though they're seagulls, but they're just so big. A flock of cormorants, which I was not expecting down there. I've only ever seen them in the Falklands before, not down that far south. And an emperor just standing by himself looking a bit sad. More Adélies trying to get away. And some of them were actually molting, which they've got to actually stay on the ice. Luckily, there was a lot of ice around, so we didn't scare them into the water. But they do need to stay until they've grown their new feathers in before they can get back into the water. And lots of those crab eaters there. This is my Pilates crab eater. Really good stomach muscles, that one. Great teeth. A little weddell. And I just love the facial expression on this guy. Looked like a pet dog, isn't he gorgeous? He's just like, what is this giant red thing that's coming right past me? And my favourite, the really lazy weddell seal. That's how I feel on a Sunday morning. Some other highlights were seeing the first iceberg. That's always a huge highlight in Antarctica. We had a visit from the Polish tern, which is down there as well, a big German ship. And we had a lot of Germans on our ship, which meant that a lot of people knew each other too, which was great. they showed off with a bit of a flyby. And Elephant Island. So this is the area where Shackleton's men actually waited for rescue for 3 months, and actually seeing it was just amazing, and to see how rugged and how difficult it would have been for them to survive there. And you can see why it was that they stayed down on the beach. There was just nowhere to go. We had some great media partners on board from the BBC, Liz and Dan there. Liz was actually she got her BAFTA for that iguana versus snakes thing that came out. Yeah, so she's just fantastic, and she actually comes from a scientific background too. She'd been on the JCR as a scientist in the past, which was great. And Dan was using new toys, lots of drones. These came in very handy when we were trying to find our way out of the ice. And one of my favourite was getting up for sunrise in Prince Gustav Channel. Just beautiful. The colours were just amazing. See the 2 little seals down there too? But pictures don't really do it justice. So if you haven't been down there, I hope you get to go sometime. And thanks very much for listening. Happy World Environment Day. Couldn't help it.